Regulation. Thank you. Dear colleagues, dear everyone present, today is the 2nd of February 2021, 12:00 Moscow time. Let me start the Dissertation Committee session dedicated to the dissertation defense of Ilya Evgenievich Borisenko's dissertation for a degree of Biological Sciences in the degree field 0.5.23 Developmental Biology and Embryology on the topic regeneration of sponges, Halisarca, dujardinii, class Demospongiae, and Oscarella lobularis, class Homoscleromorpha, cellular mechanisms and participation of Wnt signaling pathway. By the act of the Academic Secretary, St. Petersburg University, Alexander Gnatov, I was appointed chairman of this dissertation committee. My name is Andrei Ostrovsky, a Doctor of Biological Sciences, Professor of Department of Invertebrate Zoology at St. Petersburg University. Also in the panel, there is Elena Evgenievich, Doctor of Biological Sciences, Head of the Laboratory of Comparative Physiology, Developmental Physiology. Can you see and hear us? No. Yes, Igor Yurievich Dolmatov, Doctor of Biological Sciences, leading research fellow of National Academic Center of Marine Biology, named after Germonsky. Can you hear and see us? Dr. Bernard Michael Degnan, PhD, Queensland University, Australia. Can you hear and see us? Yes. Yelena Nikolaevna Temiro, Doctor of Biological Science Professor of the Department of Zoology of Invertebrates, Moscow State University. Unfortunately, she is absent for an excusable reason. And also, the candidate is here, Ilya Evgenievich Borisenko. Please, Professor Degnant, you should keep your camera on all the time. This is the regulation. And also the academic advisor, Alexander Vladimirovich Yerisovsky, Professor of the Department of Embryology of St. Petersburg University. To improve the quality of communication, please, those colleagues who are here in the room, switch off your mobile telephones, whereas those people who are online with us, keep your mobile telephones in silent mode, but switched on for emergency communication. And I'd like to inform you that an audio-video recording is on of the and it's broadcast live on St. Petersburg University website. The audio-visual recording is accompanied by simultaneous interpretation between Russian and English. Also, on the website of St. Petersburg University of this session, there's an email where anyone can send questions, and that will arrive, will be available from the technical services, and they will relay to me. But the questions should be signed, should have an affiliation of the author, and should have immediate relation to the academic discussion or the dissertation, and present an assessment of dissertation or question. Anonymous questions or questions with no relation to the academic discussion will not be read. And in accordance with the procedure of awarding academic degrees at St. Petersburg University of Candidate of Sciences and Doctor of Sciences and I will use the word procedure thereof. Hence, the work of a committee is deemed legitimate if not less than 4 people are present, which is not less than 2/3 of the established membership of the committee. We have 5 members on the committee, and 4 are present. So, including 3 members in the online mode. So therefore, the quorum is there. And I would like to go now over the agenda. The first short presentation of the chairman about the paperwork that has been submitted by the candidate. 5 minutes. A short presentation of the candidate of not more than 15 minutes on the key points of the thesis. Third, questions to the candidate, strictly on the presentation, not more than 2 minutes per question. Answers of the candidate, not more than 5 minutes for all. And then members of the dissertation committee will take turns presenting their assessments following from their reviews, and then the chairman will also have 10 minutes each, and then 10 minutes for the chairman to do the same, and then 20 minutes will be allocated for the answers of the candidate, and then there's an open floor discussion. Anyone who would like to speak up, not more than 2 minutes per speaker, will be able to do so, but provided they enter their name in the logbook with affiliation, full name, place of work, etc. And then also next, the chairman will read any questions that have arrived via email. Questions that will take more than 2 minutes to read will not be read. Then answers of the candidate, not more than 2 minutes per question. And then the academic advisor will have 3 minutes to talk about the candidate and the work that's been done. Then the dissertation committee members will have an opportunity to discuss the verdict behind closed doors, 5 minutes if needed. Then open roll call vote and the counting of votes and entering the result into the minutes. And then a decision, the verdict to award or not to award the sought academic degree will be taken, and the concluding statement from the candidate, not more than 2 minutes. Dear colleagues, do we have any questions or objections to this agenda? No? Okay. Then I will start with item 1. And I would like to, again, ask you to switch off your mobile phones here, or keep them on, but in silent mode. But those who are online with us. But also, I would like to keep your microphones off unless you're given the floor. Thank you. All right, I'm starting. The thesis of Ilya Evgenievich Borisenko, seeking academic degree of candidate of biological sciences in the degree field 1.5.23, but the developmental biology, embryology, on the topic of regeneration of sponges, Halisarca, dujardinii, class Demospongiae, and Oscarella lobularis, secular mechanisms of participation of Wnt signaling pathway. The paperwork has been submitted by the candidate in full compliance. The number of the publication of the candidate is 6 in the journals Index, in Web of Science and Scopus, 6 publications, and in the refereed journals from the list of approved by the Ministry of Science and Education of the Russian Federation, 0 publications, but I found 1. Anyway, all the paperwork is in the personal file of the candidate, and copies of which are available from a staff member of the Office of Dissertation Council Support, present here. Dear members of the Dissertation Committee, do you have any general questions to the candidate? Do we need to read the whole list of documents that has been submitted? No need. Okay, thank you. I hope that, yeah. Thank you. Okay, Ilya Evgenievich, please prepare for your... Thank you very much. Dear Chairperson, dear members of the Dissertation Committee, dear listeners, online or present here, I would like to present to your attention a report that outlines the main results of my dissertation for the degree of candidate of biological science. The problem of regeneration mechanism has attracted the attention of biologists of areas since the description of this phenomenon several centuries ago. Many animals have the ability to regenerate reparatively, and the scale of these activities varies. At the level of molecular mechanisms, the regeneration of only a small number of animal species has been studied. Model species show that most of the molecules involved in regeneration are present in species with limited reparative abilities. This is one of the main questions of the regenerative biology. Why do different animals different the ability to regenerate if the sets of molecules that regulate morphogenesis are similar in all? Slide 2, you can see at the level of cellular mechanisms, custom to distinguish 3 stages: epimorphosis, morpholacsis, and transdifferentiation. And epimorphosis is characterized by increasing the proliferative activity of cells. Morpholactic regeneration occurs by rearrangement of the remaining cellular material without active proliferation and growth. Transdifferentiation, or metaplasia, is the transformation of differentiated cells of one type into another. In all, the regenerative abilities of most types of sponges are extremely high. They're able to regenerate after operations of various kinds. Extirpations, especially of small fragments of the body, cutting into parts along the apicobasal bases or across, dissociation to individual cells. Data on the molecular mechanism of regeneration in sponges have only begun to emerge in the last few years. These mechanisms are practically not described. We can only assume them by analogy with bilateria. Within the sponge time, 4 classes are distinguished: lime sponges, calcarea, ordinary sponges, Demospongiae, Homoscleromorpha, and hexactinellida. The objects of our study belong to the class of Undersponges and Homoscleromorpha. You can see that the presence of common abilities for regeneration in different animals prompts us to research for common molecular mechanisms of this process. Such a mechanism can be the wind cascade, one of the one of the oldest pathways of intercellular communication, which is involved in a huge number of developmental processes and regulates body polarity, proliferation, differentiation, migration, and apoptosis. Its participation in regeneration has been shown in many model objects of regenerative biology, including on hydra, flatworms, insects, echinoderms, ascidians, and vertebrates. Based on the above facts, we set ourselves the goal of describing the secular mechanisms of regeneration represented of two classes of sponges, Halisarca dujardinii and Oscarella lobularis, and also to study the involvement of the wind signaling pathways in the regeneration of Halisarca dujardinii. To achieve this goal, we formulate the following research plan responded to the task, and consists of two blocks, morphological and molecular. The morphological part describes the regeneration of these two types at the ultrastructural level. Assess the contribution of the proliferation to the regeneration process. 3. Assess the contribution of different cell types to regeneration. Molecular parts identify participants in the canonical beta-catenin-dependent wind signaling pathway in the H. dujardinii, Halisarca dujardinii, and describe the special pairs of wind cascade ligands and intact Halisarca dujardinii sponges. Describe the dynamics of patterns of expression of wind cascades ligands in these Halisarca dujardinii during regeneration. The plan of regeneration experience can be the same for both species, and the inner part of the body containing choanocyte chambers is called the chronosome, and cultivated in the equinarium, selecting material for the fixation after 6, 12, 24 to 48 hours surgery. The observed regeneration process in Halisarca dujardinii was subdivided into 3 stages: formulation of regenerative plug, second, wound healing and formation of the plastema, and 3, restoration of the ectosome and choanosome. 6 hours after the operation, the SEM clearly shows the boundary between the intact exopinocoderm and the wound surface. 12 hours after the operation, a blastema begins to form, an accumulation of undifferentiated cells under the wound surface. At the ultrastructural level, choanocytes lose their characteristic trapezoidal shape and the color of microvilli. A large number of cells have phagosomes in the cytoplasm. At the stages of 24 and 48 hours, differentiated blastema cells begin to form a new exopinoderm. After 72 hours, the new exopinocoderm is completely formed. A new choanocyte chambers are formed in the mesochyle. And then to assess the contribution of cell division to regeneration, we'll evaluate the incorporation of a labeled thymidine analog. And during regeneration, it was not possible to detect any increase at the level of proliferative activity. All labeled cells were also distributed randomly. Separate labeled nuclei were observed in the blastema, but no more than in the intact choanosome. In Oscarella lobularis, the following events occur in response to the same experimental injury. After the operation, the cells of the aquifer system that appeared on the wound surface, conocytes and endopinococcites round and migrate, spreading over the surface of the wound. In 24 hours after the operation, a significant part of the wound surface was covered with newly formed exopinococcites. The shape of these exopinococcites exopinococcites is varied and rarely corresponds to the shape of the intact cells. In contrast to the intact exopinocoderm, the surface of the emergent exopinocytes is devoid of glycocalyx. You will see that scanning electron microscopy clearly shows that how they gradually lose the colors of microvilli and change shape on transmission electron microscopy, both of them, and in some new exopinococcites, carcass particles and phagosomes can be observed. Similar to Halisarca, we assess proliferation in Oscarella by the incorporation of EDU into nuclear DNA. And the slide shows a transverse view of a series of confocal images where the exopinocoderma in an intact specimen or the wound surface is seen from above. Thus, we have shown that regeneration of Halisarca dujardinii has the features of epimorphosis and differentiation. Regeneration of Oscarella lobularis occurs due to the movement of two cell layers in the wound area. And then we sequenced the adult Halisarca dujardinii transcript and identified the wound cascade participants in it. The diagram shows a diagram of the signal pathway at rest and when the ligand binds to the receptor. In the absence of a signal, the cytoplasmic complex of proteins ensures the destruction of beta-catenin. When Wnt binds to the frizzled receptor, a number of proteins are recruited to the receptor and this complex is destroyed, as a result of which beta-catenin is not destroyed but enters the nucleus where it works as a transcription factor. We identified 10 Wnt ligands, 6 receptors, and all participants of the cascade except axon. Accuracy, it is axon that plays the role of a backbone in the complex that destroys beta-catenin. Phylogenetic analysis shows that none of the ligands showed sufficient affinity to the Wnt families described in the in other animals. Apparently, Wnt in sponges arose after the formation of the type from the last common ancestor, and expanded already within the type. Using in situ hybridization, we visualized Wnt expression in adult sponges. Larvae regenerates in order to assess the localization of transcript. A number of transcripts showed expression in the osculum, in exopinococytes, in peripheral exopinocoderm and basopinocoderm, at the base of the ocular eminence and the cytoplasm of oocytes, and in the larva in the posterior hemisphere and along the equator and the superficial flagellar cells. Noteworthy is such a model of regeneration as a whole-body regeneration, which is also characteristic of a sponge. Sponge cells, after dissociation in an adult animal, are able to assemble into an aggregate, primorph, which will develop into a new full-fledged sponge. We analyzed wind expression activity during pre-morph development using in situ hybridization and RNA sequencing. It was shown that 6 out of 10 ligands are actively expressed during the development of pre-morphs, and the expression of some of them significantly changes when comparing stages 1 and 6. Stages 1 and 6 days after dissociation. In situ hybridization on pre-morphs, different stages showed that polar wind expression can be observed starting from day 1 after dissociation, i.e. at the stage when there are no signs of morphological polarity yet. We have observed polar expression of different ligands at different stages of development, as well as in the emerging channels of the aquifers system. The data obtained allow us to draw the following conclusion. The morphogenic process during reparative regeneration, the Halisarca dujardinii, are based on the epithelial mesenchymal epithelial transitions, differentiated choanocytes, endopinococytes, and archaeocytes serve as sources of the formation of an accumulation of cells, a regenerative blastema, from which cells further migrate to the wound area. Reparative regeneration of Oscarella lobularis is carried out by the reorganization of intact sponge tissues according to the type of epithelial morphogenesis. Cellular sources of regeneration are choanocytes, exo and endopinococytes, which differentiate into exopinococytes. Sequences and transcriptome analysis of the sponge revealed all participants of the canonical catenin-dependent Wnt cascade, except for axon. The expression of Wnt genes in Halisarca dujardinii is observed during the apicobasal axis of the definitive sponge, as well as along the anteposterior axis of the larva. Expression of one of the ligands, HDU Wnt K, is observed at the border of the wound surface and the intact Halisarca dujardinii. And 6 components of the Wnt signaling pathway are expressed in different stages of regeneration during the development of pre-morphs from Halisarca cell aggregates. Based on the materials, 6 printed works were published in Web of Science and Scopes. Thank you very much. We have now question time and reviews. Therefore, do you have any questions? The members of the station come strictly on the presentation. Do you have any questions? And I hope we have questions. I have no questions. No. I have a question, please. Ilya, could you tell me, when you talked about the Wnt signaling pathway, and you demonstrated the tree, you said that spongy Wnt emerged separately from other animals after separating the sponge from Bellateria. Do you think that Wnt genes in sponge are not homological to other animals? Yes, please. Evgenievich, no, this is not what I wanted to say, of course. What I wanted to say is that there was a limited number of homologens from the common last answer to about 4 genes. This this developed into a whole family in spongy, which in phylogenetically we cannot compare with the Bilateria family. They are homological, but the expansion developed after these very small number of ancestor genes got there. Are you happy, Igor? Yes, I am happy. From your presentation, I understood that what Ilya said. I didn't have that question. Can I ask my questions, or do we have any other questions from members of the Dissertation Committee? Bernard would like to ask something. Can you hear us? Well, can you hear us, Bernard?
Yes, I can hear you.
Yeah.
Do you have any questions?
Do you have any questions at this stage?
Yeah, the in situ hybridization protocol that you used, in the, I think it's the next slide, what controls did you run?
Понял.
I will speak Russian. We used controls as positive controls, the genes of tubulin, which gave which gave the coloring of chronocytes and echocytes, and the meaning sense some probe, and we didn't get any localized or background coloring.
Thank you.
I have two questions. Could you elaborate on phagocytosis? What is the role of phagocytosis in regeneration process? This is a question that needs additional study. What we saw in structural picture, phagocytosis plays a role in eliminating cells that had been damaged through operation. This is a massive phenomenon. Whether it plays any phylogenetic function, I cannot say. I don't know. The logical solution so to answer your question is to block some inhibitors and see phagocytosis and see if the regeneration gets disrupted, if whether it all gets reduced to the eliminate. Well, as far as I know, it's the reduction, the elimination of damaged cells. Chronocytes in Halisarca, in both types actually, are the most numerous, and of course the mesenchymal Halisarca cells as well. For another short question, you showed the expression of Wnt. Is this very characteristic of spongy for spongy, but also maybe a scarcity of bilateria as well. Well, they're comparable in bilateral symmetrical animals, but we never saw expression in the front pole. And there's another question here. Can we compare the front and the back axis? On the one hand, yes, they're similar, but on the other hand, dissimilar. Thank you very much. Well, it's understood that nothing is understood, but do you have any other questions? Well, I have a question. Yes, Irina Evgenievna, please. I have some reviews, some questions in my review. Can I read some of my questions from the review, because they're theoretically, yes, you can read your questions now. Well, the microphone, is it on? Your microphone, is it on? We cannot hear you. Yes. These questions are, well, very good experimental material. So many experiments you did, morphological, removing some parts, and have you counted the morphological changes? As we talk about structural part of the work, we took a minimum of 5 regenerates for each stage of the study. This is a minimum of 5 samples for every temporal point, 5 for scanning and 5 for marking DNA, and 5 because we need some statistical proof, but some samples of course get lost. 5 is the medium, golden medium, and it's enough. Sequencing is a part. What's the percentage of cases? Maybe 100%. The changes that did occur, because there are very many different conditions, and did you have changes more in some, less in others? The diversity is determined by the scale of injury, whether you cut along the sponge and take a small fragment of body, and there will be no total destruction there, but 1 of 5 samples actually dies for some reason. Morphological changes occur in all cases. This is a standard program. So what's the the percentages there? Well, it's a small critical remark. You don't have enough probes, and I think, and not enough calculations done, and I would have loved to see more figures, numbers. well, you said that dissemination was not equal. Well, the chambers you can visualize conates, but you have to subdivide it into different injuries should be differentiated. Do you see no difference in the type of injury? This was not our goal to quantitatively treat, because this was a voluminous work. Well, I did that after a paper. The paper was published, and unfortunately, dissertation, as far as I know, cannot include unpublished material, and so in the radius of half millimeter, there's no activation. Well, it falls, can fall, but statistically it's not proven. So activation, we think that activation doesn't happen. And type of cells. We talked about different types of cells. They're all involved in the regeneration process. How do you identify these cells, different cells, morphologically? First of all, conocytes, they keep the basal body for a long time, and there are a lot of nuclei, ambocytes. They can be. You can recognize them by a nuclear in this, and these latter ones don't participate in creating a blasteme. They accrue phagosomes in cytoplasm, but these are morphological markers that we used. Thank you. Thank you very much. Maybe while listening to the answers, you came up with questions. Any more questions? No? Okay. We have time for reviews, of course. Time to compensate. Now, members of the Dissertation Committee will have time. Will have the floor. So questions, critical remarks, and external reviews. So I would like to remind you that the candidate can answer questions and critical remarks after each review, or in full after all the reviews have been read. Maybe it's easier to answer after each review, but so therefore we'll stop after each reviewer, and since all the reviews have been published on the website and are accessible, I suggest that you read the key points only, including the critical remarks and questions and suggestions. Well, of course, you're entitled to you may feel free to read the whole review, but to save time. So the first from Professor from Moscow State University, Elena Nikolaevna Temereva. So, regeneration of regeneration of Halisarca, dujardinii, and Oscarella lobularis. So this is the review from Elena Nikolaevna Timirova, Doctor of Biological Sciences, Professor of the Russian Academy of Sciences, Professor of the Biological Faculty of Moscow State University. I would like to read it. The regeneration is a vital process that is expressed in all living organisms as the regeneration of parts of body and recuperation of wounds takes place in all animals by similar ways, but they are able some are able to regenerate parts of the body and others only to heal the wound, regenerate the wound. The reasons for such differences are not known as of yet, and as of now, and in this connection, a detailed study of cellular and molecular mechanisms of regeneration in representative of different parts of metazoa is very relevant. And the work is structured according to a standard design that consists of introduction, etc. And therefore, I would like to go to read the critical remarks. Well, there is no description of the general morphology of Halisarca and the journey, and the dujardinii and one scalumna or monogascalum. Is it in the manuscript it says that the spongia is of a smooth form. This description is wrong. And I would like to say that some parts of the literature view use everyday language, and the author uses to describe hydra such words as head instead of the oral end of the body, a leg instead of the foot, and so on. And second remark is about the phrase that the spongia doesn't have organic and non-organic skeleton. This is a strange phrase because all spongia have organic skeleton, spongia or collagenic collagen. Number 3, from the descriptions of both spongia, we have confusion. What do we understand under ectosome for the first Halisarca dujardinii? This is a very complex 3-layer structure that includes exopinacocytes, but Oscarella lobularis, for its ectosome is represented by thin, from 10 to 50 micron layer of mesochile, and the exopinacoderm is not included in the ectosome. Number 4, to understand the text, for better understand the text, you should have been more clear about the terms. What do you use the term mesokill in the VKM and SEM, and this has to do with the legends and the figures. So the use of the term basal membrane instead of basal plane is not right. These two notions are different. Chapter 3, which is part of the results and discussion, is not well structured. It starts with a literature review, which then turns into the description of data, interspersed with citations of or the references to literary sources. And number 7, some data about the distribution of wind cascade genes received on Halisarca dujardinii. Can they be related to Multiscleromorpha? And so the dissertation, as far as the section material and methods, you have to. This is too general, and we need more detailed information. Anyway, but on the whole, the work of Borisenko, Ilya, Evgenievich is a complete work, and the commending part of this dissertation is the experiments and the description and the control of the experimental data, the results, and so on. And you have, in this work, there's a comparative analysis of the results which are obtained for 2 representatives of periphery, and also the general results of the work and literary sources. The conclusions that follow from the work completely match the findings from the experiments. They're relevant and original. So the general conclusion is that the thesis of Ilya, Evgenievich, Borisenko on the topic of regeneration of Halisarca dujardinii and Oscarella lobularis cellular mechanisms and the wind signal cascade meets the key requirements of the procedure of awarding academic degrees at St. Petersburg University, and the candidate Borisenko Ilya Evgenievich deserves this said degree to be awarded to him in the degree field of biology, developmental biology, embryology. No violation of clause 9 and 11 of the procedure. This is a very critical analysis. I would like the first of all, I would like to answer as far as the I will answer first the comments that are separately indicated in the review and then the general ones. Halisarca dujardinii may have one or more osculums, but at large individuals are usually multi-osculum. Slide 23. The shape of the body of a sponge probably depends on the hydrodynamic regime and the substrate on which it lives. The shape of the sponge is both cortical and scale. By smooth was meant the surface of the sponge, because in other common species, such as the Helicondria panicea, it is uneven. Second, I agree with the review that mesohyl also performs skeletal functions even in the absence of spiculus. Well, it's very bad formulation, yes. E3. In the Halisarca, the exopinococytes are T-shaped, and although their caps lie on the surface and form a cover, the cell bodies with nuclei are immersed in the mesocyle. By definition, the ectosome is a layer of a mesohyl between the choanosome, the inner part of the body with an aquifer system, and the exopinocoderm, in which there are no choanocyte chambers. The exopinococytes themselves are not part of the ectosome, included in Halisarca, but the cell bodies are immersed in it. 4. Mesocyle includes the extracellular matrix and populations of resident cells, archaeocytes, spheral cells, granular cells, lymphocytes, etc. Thus, the extracellular matrix is a mesohyl component. 5. I acknowledge the incorrect use of the term basement membrane, because in English, the English language literature, the basal membrane is usually used in relation to the basal plate. The same is true for the review of literature, where many words are familiar from foreign literature, for example, head and foot in Hydra instead of hypostome and soul. Number 6, with regard to the general structure of the work, which is generally noted in the review, I can explain the following. On the whole, its structure corresponds to the state standard, the national Russian standard, but moving away from the usual organization of the text, I'm moving to the end of the materials methods, combining results and discussions with data from a literature review. I relied on papers previously defended according to the rules of St. Petersburg University. The trend towards a reduction in the overall volume of work and a different structure of the text was the intention, as far as I understood, to simplify acquaintance with the dissertation and thus increase the number of readers. So the reader will be able to get acquainted immediately with the next work of the author without studying all the articles. Maybe it's not the right decision. As far as the functions and functions, we're still far from compiling any complete picture. The diversity of ligands, the diversity of ligands within the phylum, the absence of their phylogenetic relationship with bilateral bilaterian ligands, and the lack of functional data raise more questions than answers. We are working now on cyconcyllatum, and we which is located. It is clear that the osculum in which some winds are expressed is located at the end of the body axis of the adult sponge. The method, as with regard to, so theoretically it can be called apicobasal for Halisarca, and another multi-osculum sponge, Leucosolinia. We also showed the expression of some winds and osculums. The Sun published it. With regard to the methods, I explained that they are all described in detail in articles which are referenced. electron microscopy, including sample preparation for tissues, was carried out by the applicant as indicated in the section personal contribution of the author. Yes, thank you very much. I hope that the reviewer will be happy. Terminology problems are a very weak part because foreign terminology and Russian terminology don't match in many respects, and most young researchers use foreign terminology, and the scientists of our generation grumble that terms are different, the names for the terms are different, etc. So some questions have been asked. Please read the last part, which is a formal part of the quality of the dissertation. Thank you very much. Can you hear us? Can you hear me? I would like to say that I was very interested in the work, a very relevant topic. The structure is standard, and the previous review said that a lot of original experimental data is presented, so the work is very valid. The validity of the provisions in the conclusions is there. I would like to say that the work is presented in 2 languages. This is very good. Both texts are readable, and so this work is done at a high level, including methodology and contemporary approaches. I already said something about experimental material, morphological. Well, I already asked my questions about that. I would have liked to see more illustrations, but and additionally, you should have marked basal apparatus by gamma tabulating, and then you could have calculated to increase the weight of the results. Then I would like to see more differentiating dispersion of the 2 different spongi, and to see why you chose these 2, and I would like to that the work would have been better with more figures, especially about the contemporary understanding of structures, molecular structures of the spongi, and then you would have I would like to see more when you describe the process of morphological changes of the specific types of cells, you talk about the changes of form, but yeah, so it would be good to see photographs of one of the same type in the normal condition, and to compare what's happened to them at different stages of regeneration. And in the work, you have to provide a list of used abbreviations, because not in all cases do you use a full name before you use the acronym and or abbreviation. Well, anyway, and also I would like to say that it's evident that the work uses two languages, so I would like to see some of the phrases to be translated in literal Russian instead of just transliteration or calc translations, to match with the terms we are accustomed to in the Russian language. Nevertheless, on the whole, the work meets the requirements of the procedure awarding academic degrees at St. Petersburg University, and Borisenko Ilya Evgenievich deserves the third degree of candidate of biological sciences to be awarded to him. Thank you very much. Evgenia Voronezhka, please. Ilya, please answer. Of course, I agree with the remark that more figures should have been provided to facilitate the reading. And as far as the calcs from other languages, also I admit that these are my drawbacks. As far as the use of two different classes, we took representatives of two different classes to see how similar different morphogenesis would be in phylogenetic distant species, because yeah, used to be the second sponge was part of the Halisarca, but now they're separated. For example, Homoscleromorpha were previously included in class of ordinary sponges along with Halisarca, and now they're separated into a separate evolution and more advanced class. We found, as expected, that in them the integrity of epithelial structures during regeneration is preserved to a great extent than in the Demospongiae sponge. The next review by Professor Dalmatov. Please, do you have any? You can read the full. I study something similar and enjoyed reading this dissertation. I'd like to stress that upfront that there are many commendable properties in this dissertation, but the main thing is the comparative analysis of two classes of periphery with different organizations, and therefore that led to very interesting properties and compare. This is very important for in evolutionary sense, to have in basal forms of animals two different types of regeneration, and and that sheds light on this phenomenon, and the author demonstrates very interesting findings. As far as my critical comments, I would like to side with the comment of the structural drawbacks, methods at the end, and results front. Why do you have the chapter literature review, and then there's no chapter results and discussion? And because I know this material quite well, when I read this dissertation, I saw the drawback of this work is the conciseness in the description of the results, very telegraphic style, and a lot of the important properties are left out, and they're very difficult to, if they are mentioned, difficult to take them in. As far as the exopinococytes in the regeneration of Halisarca, I would like to hear more elaboration of this. What did you mean? So what's the role of them? Why does the author think when the regenerative membrane, when was the regenerative membrane created, and where did it disappear later? If it's as it says in chapter 1, regenerative membrane is a temporary formation, and how does and then other questions, was there a fate and the function of endopinococytes that appeared on the surface of the wound 6 hours after the injury? How do the process of formation of regenerative membrane and the migration of exopinoderm, how are they compatible, and to which stage and how do the endopinococytes integrate into the exopinoderm? So also the dissertation has typos and grammatical mistakes and some inaccurate translations of English terms. Nevertheless, these drawbacks don't diminish the value of the revised work, and therefore the dissertation of Ilya, Evgenievich, Borisenko on the regeneration of Halisarca, dujardinii, and Oscarella tabularis, cellular mechanisms, and the Wnt signaling corresponds to the procedure of awarding academic degrees at St. Petersburg University, and therefore the candidate Borisenko Ilya Evgenievich deserves the third degree of candidate of biological sciences in developmental biology and embryology to be awarded to him. Thank you very much. Thank you very much, Igor Yurovich. very close reading of the work when a specialist can shake the soul out of the work. As far as the structure of the work and the name of the chapters, I tried to shorten to cut the volume of dissertation to as it was specified by the regulations of St. Petersburg University, and therefore I focused on recently defended works. The rules do not provide for requirements for the section materials methods results in discussion, so I found it possible to do it in the manner of published dissertation. The confusion about exopinacocytes is due to a semantic error in the text. Exopinacocytes of Halisarca, in our opinion, are terminally differentiated cells and are not directly involved in regeneration. The shape of the definitive exopinacocytes, T-shaped with the cell body immersed in the mesocycle, does not allow them to leave the exopinacoderm. Indeed, 12 hours after the injury, the layer of the exopinacoderm near the wound dissociates, but we did not observe the release of exopinacocytes from the integument. Accordingly, exopinacocytes are not part of the blastema in any form. In the course of regeneration, as new exopinacocytes are formed, differentiating from blastema cells, the boundary between the wound surface and the intact exopinacocytes disappears, filled with new cells. As far as I know from the literature, the presence of microvilli is one of the signs of cell activity, including motor, although mobility is one of the forms of activity. As far as the epithelialization of the wound in Oscarella occurs as follows. After damage to the wound surface, three populations of cells border the surrounding exopinacoderm, endopinacocytes of the cut channels of the aquiferous system and the canococytes of damaged sinusoid chambers. Coenococytes undergo transdifferentiation at their location, and the exo and endopinacocytes begin to move. Accordingly, the endopinacocytes move outward from the channels. Exopinacocytes move from the periphery to the wound surface. And the coenococytes remain where they ended up as a result of damage to the coenococytes chamber. And all three cell populations gradually fuse to form a new exopinacoderm. The mention of a regenerative membrane is unfortunate here. It meant a new exopinacoderma. Igor Yurich, have I answered your questions? Yes, you have. Thank you. It would be interesting to continue the discussion, but not maybe in this context. Well, there will be a question, there will be a discussion time during the session, so if maybe we can continue there. In this context, based on your questions, what do you mean by pinacocytes move? Endopinacocytes move. do they split? Do they crawl? What do you mean by moving? As you can see in the series of temporal points, we see that gradually the layer of cells move. No, they don't split. They move constantly so cells can become flatter, and the area can change, and they can change position. Form of cells, yes, that does change. As far as the position, I'm not sure. Theoretically, yes. So, thank you very much. We have the next review from Degnan. Dr. Dector, I would like to give the floor to him.
Thank you. I thank the chair and the committee for inviting me to assess this thesis of Ilya Borisenko. I do not know much about the process at St. Petersburg University, nor the procedure, so I will not comment on the structure of the thesis. I will give an overall assessment and then maybe go to a few questions related to specific topics in each chapter. So I think Ilya Borisenko presents a novel and interesting research into the cellular molecular basis of regeneration in a Demosponge, Halisarca, and Homoscleromorpha, Oscarella. The molecular work focuses on the Wnt signaling pathway in the Demosponge, and as far as I can tell, this is the first study to look at the expression of Wnt in an adult Demosponge and primorphs and in the process of regeneration. So in that sense, the thesis contributes substantially to our understanding of regeneration in these two species of sponges and provides an important foundation for comparative research into the future. The English version of the thesis, I cannot comment on the Russian version, is in general well written and presented and scholarly. The results, figures, and figure legends are well presented and include high-quality scanning and transmission electron micrographs, confocal micrographs, and whole mount in situ hybridizations using specific Wnt probes, and includes informative schematic diagrams, as shown right there. So I have a few comments and questions I would like to ask, and I will go through them systematically through the thesis. And I will start with the introduction. And one issue that comes up often in the thesis is the terminology transdifferentiation and dedifferentiation. Often used to talk about choanocytes dedifferentiating. And of course, dedifferentiation is a subset of transdifferentiation. So do you mean dedifferentiation? And if so, what are the choanocytes dedifferentiating into? This would have to be a previous cell type from which the choanocytes emerged. Or is it just transdifferentiation? And so I would like to ask you a theoretical question based on your literature in relation to the relationship between the diversity of cellular regenerative mechanisms. For instance, cell proliferation occurs in some species but not others. Yet there appears to be a consistent and conserved use of Wnt signaling in all regenerative processes. My question to you is, how do you interpret this? Is it that Wnt has been co-opted multiple times? Or do you believe that Wnt is an ancestral deep conservation? So moving on to the second chapter on cellular mechanisms. Again, I thought this was a very strong chapter and did a good job in combining electron micrographs with EDU labeling. I do have a few potential questions for you, though, that might be worth thinking about. You propose that the choanocytes migrate to the wound and then transdifferentiate based on your transmission electron micrographs. Do you have direct evidence that the choanocytes are actually transdifferentiating? For instance, do you follow EDU labeled migratory choanocytes and observe transdifferentiation? A second question is, in Oscarella, you used Indian ink to label choanocytes, and you show that they transdifferentiate into exopinacocytes. And that's an excellent experiment. Did you see any evidence of an intermediate cell type between the choanocyte and exopinacocyte? So moving on to the Wind chapter, chapter 3. Again, I thought this was good pioneering work, excellently done. And I have one theoretical question from this chapter, and that is, you mentioned the existence of a Wind code to pattern axes of cnidarians, ctenophores, and sponges on page 151. Are you willing to propose how such a system works? And more importantly, if the code in these taxes are homologous? How do different winds provide different information to receptive cells? So those are the questions that I have for you. So in conclusion, I think that this dissertation by Ilya Borisenko for the degree of Canada of Sciences in Developmental Biology and Embryology, thesis titled Regeneration of Sponges, Halisarca, and Oscarella, Cellular Mechanism Participation with Signaling Pathway, appears to me to fulfill the needs, the goals of a thesis in most places in the world. Thank you.
Большое спасибо, доктор Дегнан. Очень детальная, очень интересный отзыв. Илья, пожалуйста, это будет не очень просто.
Я, с позволения, буду отвечать вот точно по порядку, как...
I will answer the questions in the... I will not read the question, but I will just answer, will read my answer. As far as the irreversibility, no, definitely not irreversible. All everything is determined by whether the target type of differentiation is terminal or not. For example, Halisarca exopinacocytes are themselves terminally differentiated, so transdifferentiation in them would be irreversible. Choanocytes, on the contrary, are plastic in this regard. Therefore, if other cells, amoebocytes, for example, are included in the new choanocyte chambers, then such transdifferentiation will be reversible. Secondly, taxon, the sponges, the most ancient taxon of multicellular animals. I totally agree. Sponges may not be the oldest living phylum of multicellular organisms, but they certainly are one of the earliest phyla. The statement from the introduction is careless. As far as the... Yes, and here we have the gistology of Halisarca. The second, as the choanosoma is number 2, as the internal parts of the cell, and there's one on page 120, no reference. The data given in the monograph was obtained by the applicant and published in article number 137 from the list of references. The lack of a link in the paragraph is my fault. As far as the unclear what the ECM is demarcating in panels A and B. In figure 3, the letters ECM represent the intracellular matrix. On panels A and B, the extracellular matrix of the ectosome is just visible under the caps of exopinacocytes deformed during sample preparation for electron microscopy. As far as the page 131, this temporary structure contains differentiated cells such as this. So, this is an inaccuracy in my translation from Russian to English. It is implied that this temporary structure contains de-differentiated cells derived from choanocytes, endo and exopinacocytes. In addition, archaeocytes are included in the blastema. So it talks. So that as far as the last question, very important question. Identified Wnt expression domains that completely cover the apicobasal axis without Halisarca. As far as we can judge from the data on the cellular composition of the definitive sponge, yes, the statement is true. For example, the expression of the Wnt series is observed in the exopinacoderm, and the exopinacoderm at the border with the basopinacoderm does not differ morphologically from those at the level of the oscular tubercle, but express different Wnt. Morphological. More data on the activity of the cascade would, of course, be obtained by studying the localization of beta-catenin by immunochemical methods, but we have not yet achieved success with antibodies. Thank you. Are you happy with the answers?
Yes, thank you. Yes, thank you.
Yes, thank you. Thank you very much. I would like to make a comment for the Russian. Dear members of the Dissertation Committee, we are used to different rules of defending academic degrees, but Moscow State University and St. Petersburg University have modified their defense requirements to fit international standards, and therefore the dissertation author followed the new rules, and in terms of the structure of the dissertation, in terms of the rubrication and the volume of the dissertation. We are used to bigger volumes and different designs of the dissertation, but the thesis is a complete work, so this is clear. I would like to read my review now. Of course, I would like to ask this question before this. Do we have any questions from the internet? No. Okay. I would like to, if people are listening to us, since it's live, broadcast live, then I will read part of my review, not all of it. I will not read the trivial parts, but I would like to briefly comment on the structure of the contents of the chapters. I enjoyed reading this session in relation to the works of the academic advisor, who published 2 books by and formulated very interesting hypotheses. And there are very well argued basis. Different animals, different types of animals. Some of these moments, some of these statements, surfaced, and this dissertation is very relevant. Chapter 1 is divided into 3 parts. The first one is cellular and molecular mechanism of regeneration in invertebrates. It's devoted to the discussion of regenerative morphogenesis of invertebrates and cytological on psychological and molecular levels for each type of animals. A spectrum of known regenerative process and their participating molecules are discussed, and the reader receives an understanding of the relevance and the state of the art of the research in this area. And the second part is literature review. The author discusses the wind cascade and the regeneration of model objects. And chapter 2, cellular mechanisms of regeneration, Halisarca and Oscarella, which are representatives of different types of spongiae. The author provides original experimental data on the study of process of reparative regeneration in these 2 types of spongiae. And chapter 3, the participation of a wind pathway in the axis specification of the axis of spongiae and regeneration as a central part in the dissertation, and the visualization of the expression of genes is augmented by the data of sequencing of RNA, which allows to establish quantitative differences at different stages. The conclusion summarizes findings. All of these chapters have regional data, and this is very important for the dissertation. And this binds them together. And this is a very important attribute now. Attribute of dissertations that are defended at St. Petersburg University. I agree with the previous reviewers' remarks. Unfortunately, the conclusion is all too short, and there's a dissonance of the contradiction between the conclusion in the phrase the source of cells including for regeneration is in this stably proliferating population of conocytes, and then it would be interesting to hear an explanation on this account. In the materials and the methods, you have a short presentation of the methods that are used in the work, and I would like to the work is quite logical. is based on the findings already published in the works. Some typos, some unfortunate misprints. Well, there's marked and unmarked. Then for in the Russian spellings, the same term is written in two different ways, and this is very strange, of course. And so as far as the originality and the validity of the obtained data, and also the validity of the conclusions don't generate any doubts, and it's also supported by the high level of journals where the findings have been published. And the final conclusion of my review is that the thesis of Ilya, Evgenievich, Borisenko on the regeneration of Halisarca cellular lobularis meets the key requirements of the procedure of awarding academic degrees in St. Petersburg University, and the candidate Borisenko Ilya Evgenievich deserves the academic degree of candidate of biological sciences in specialism and degree field 1.5.23 developmental biology to be awarded to him. Plagiarism points 9 and 11 items have been met, not not violated. Okay, thank you very much. So we continue. I would like to you to answer of a couple of my. With regard to the contradiction between the conclusion and the conclusions to the main conclusion and the conclusions to the chapters, I explained the following. The reference to regeneration here is hypothetical. It was said that sponges, animals with a high level of cellular plasticity and the ability to and trans differentiate, should have a population of constantly dividing cells that ensured body renewal. Conocytes are the most likely candidates in both cases. The remaining cells of the regenerate serve as sources of cellular material in the regeneration of both species. We did not find the contradiction of proliferation. As far as certain evolutionary conclusions based on the data of the wind cascade are given in the work, yes, they are given. For example, the phylogenetic position of ligands is determined. We have shown at the time it was the second work after the article by Lening et al. On second atom that the wind proteins of sponges and the Ballateria are two different groups. Such diversity in sponges arose already after the separation of the peripheral type. Moreover, if we look at the sponge wind phylogenetic tree on the slide, we can assume that a number of duplications already occurred within each of the classes. Sequences of calcarea are shown in yellow, Demospongiae blue, just a second, Demospongiae in blue, purple and green, Homoscleromorpha in red. And in this regard, it is difficult to draw, and well, you can see that there's a lot of there's expansion within a class, very goal-oriented. It is difficult to draw analogies regarding their functions with bilaterally symmetrical animals. A clear common feature is the patterning of the body axis. And here we return to the question of axis and sponges. And the larvae have an obvious morphologically pronounced anterior-posterior axis. How it correlates with the axis of an adult animal, especially a multi-osculum one, we do not yet know. But at both stages of the life cycle, it looks very likely that wind is involved in the specification of the axis. Some of the ligands may be separatively involved in some regeneration process, thus using hybridization. This one wind is not expressed in adults, only in one temporal, in adult species, only in one temporal point. The most important question in Russian, mechan and mechan, marked and marked different. There's a difference in spelling between participle and adjective. They're spelled differently. Well, there's a difference between them, and I am very sorry that I confused these two. Well, if not for the COVID, I would have had a full room, and I would have asked them, the audience, to comment. But unfortunately, now the defense is largely in an online format. Almost the dissertation committee is in self-isolation, and so therefore we have no one to ask questions really, and I think that we've had quite a few very interesting questions from the committee members already. So. And I think they reveal future research avenues. If we have any additional questions from members of the dissertation committee members, especially on the answers of the dissertation council to our very sly questions, then if we do have that, please come up with. But if not, then the academic advisor will have the floor, and who will talk about the candidate himself and how the work proceeded. Well, you don't need to wear a mask when you speak. Dear Chairman, dear members of the committee, dear candidate, as far as I understand, the advisor is not to give assessment of the work, only the assessment of the author. It's hard for me to talk about the abilities and successes of Ilya, because we've been working with him since 2011, when he just joined the university from first year of bachelor study, and he chose embryology. And even before he entered the university, we went a field trip to the Black Sea, and this is how we met. And very soon we published a joint article. Ilya then switched to Spongiae, and this was just like honey on his lips. He is the disciple of the school founded by Professor Korotkova, and it's good that this tradition, scholarly tradition, continues. Ilya for the time of his study and then work. Now he's a teaching assistant in the department. He's done a lot of work in research, and this activity sometimes irritated me because this didn't coincide with my plans. And every academic advisor wants their mentee to work according to his plan, but he deviated. But those deviations enriched his research, and he delved into molecular biology. He studied the development of regeneration in spongiae, and now, by conquering bioinformatics, we can get to a higher level of study. Ilya hasn't deviated from the classical methods, morphology, structure, these basic elements. Without them, you cannot assign any expression of genes to anything, and otherwise there's no stages. There's just it may end up on just beautiful phrasing, but so this keeping the structure in and not deviating in his self-study from the basic science is very great. He's done a lot of work in experimental work, participated in some lab experiments. He knows how laboratories work in Sweden, in France, in Norway. This helps him not only to study but also develop strategy of experiment. And he participated in different congresses, international congresses, as just listener, but also presenter, and this enriched him a lot as well. It's very this is an important element of the growth, personal growth of a researcher. And Ilya also practically, he participated in practically all the grants that I've received, not only as a member of but also as a manager of a particular area, and leading a team, and then some of these findings became part of the dissertation and publications. Again, this enriched him as well as a specialist, because this is easy to follow the advice of the research advisor, but it's much more difficult to establish contacts with companies, with research laboratories, and do something else. And as a result, we have a specialist not only in the study of spongiae and the regeneration and morphological, but a researcher who can be a team leader, a project leader, and organize his own research team. And I hope that the department has received a very good specialist, and the university a new specialist of an international standing. And emotionally, I'm very happy that this marathon is coming to an end, and I'm just very. I just have fun working with this person, And I hope, again, candidate of science, Borisenko. Thank you. I hope that your tender attitude to the to Ilya will be fruitful, because you've really brought him up as a scholar. The number of publications. The number of publications is much bigger than we see here in the reflected in the work, in the thesis. So there have been some recent works that are outside the scope of this research. And since we have no one willing to speak up, the absence of the presence of the audience, due to the absence of the presence. Do we have, just again, standard question, do we have any emails? No. No comments from the online space. So the discussion and the accepting of questions are over. And before we conclude, before the open roll call votes, we have I have a question for the members of the Dissertation Committee. And also the candidates, do you have any objections as to the procedure of this Dissertation Committee session, including technological? You have to say yes or no. I have no objections. Everyone, no objections.
No
No
objections.
objections. Нет.
Переходим к следующему.
So the next item on the agenda is the vote. Before we do that, do we need a technical break to discuss our verdicts behind closed doors? 5 minutes break can be allowed, and the broadcast will be switched off, so everybody who's in the room, apart from the members of the Dissertation Committee, will have to leave the room and so on. Do we need that? And after the break is over, do we need that? No, I don't need it.
No, I don't need it.
Окей.
Thank you. Excellent. Everything is clear, so no problem. I don't need any break. It's all clear to me. I agree with you as well. So we can proceed to the voting. I will ask every member of the Dissertation Committee to say their opinion, how they vote. I would like to put to the vote the resolution of awarding Borisenko Ilya Evgenievich an academic degree of Candidate of Biological Sciences is in the degree field 1.5.23 Developmental Biology and Embryology. I would like to say that by voting yes for this the resolution, you admit or confirm that item 11 in the procedure, which states the dissertation must refer to every source of borrowed materials or specific results, whether used in someone else's work or in his own work, in their own work, previous work. This should be specified in the work. So that you agree that this has been done. And so we start our voting procedure. It is 2 p.m. On my by my watch. Member of the committee, Elena Evgenievna Voronezhko, what's your opinion? I agree with the decision to award the candidate with the degree of candidate for biological sciences. Igor Yurievich Dalmatov, member of the committee. I also agree. I will endorse the awarding of the third degree to candidate Borisenko, and a member of the Vernon Michael Degnan. Your opinion?
I agree that the degree should be conferred to Ilya Borisenko.
Большое спасибо.
And now I also vote as chairman of the committee. I agree with the fact that the Borisenko, Ilya Evgenievich deserves the third degree of candidate of biological science. And now I would like to voice the results of the vote. Out of 4 participating members of the Dissertation Committee, 4 voted in favor of the resolution to award the third degree. No abstention, no one voted against it. So, therefore, the decision to award Ilya Evgenievich Borisenko an academic degree of candidate of biological sciences in the degree field 1.5.23 developmental biology and embryology has been taken. Congratulations. Concluding statement from the candidate. It's a very emotive moment. I'm very thankful to all members of the Dissertation Committee, and I'm very appreciative of you finding the time to participate in this event. I would like to thank members of the Office of the Dissertation Committee and the interpreter, because compared to the previous procedure, having an interpretation facilitates our work so much that we can invite members of from abroad, and I would like to thank the organizers, the staff of the Research Park of St. Petersburg University, and also the field base Belomorska. This is our field station. I would like to thank all members there, and 2 laboratories, staff of the 2 laboratories in Bergen and King Berkelini in Marseille, and I am very thankful. And the Russian Basic Research Foundation, who funded this, and the Russian Basic Research Foundation, Russian Science Foundation, and Russian Basic Research Foundation. And I would like to thank the people who have been supporting me all along, near me. I would like to thank my wonderful research advisor, who, besides the supervision of my research work, has become my role model, has done a lot to make my scientific life as successful as it has become. Thank you very much. Very touching concluding word. Thank you very much. Congratulations. This has been a long road and a lot of work. Yes, you can, congratulate Ilya, but our session is thereby closed. Thank you very much for everyone, to everyone who participated. Thank you.